All right, so today we're going to talk about how to sample solution spaces and characterize them. It's a very practical way of looking at them and studying their properties. So I'd like to start out by showing this phylogenetic tree of the COBRA methods. As you know, there are many of them, and there is one branch in this tree that says unbiased methods. And that's basically what we are covering in part 2 of this class. And the unbiased methods come down to finding the basis for the space of interest, and we talked about that last time. And today we're going to talk about uniform randomized sampling of the entire solution space. I think you can probably view the effort to get a useful basis as being more conceptual or theoretical and kind of deeper in nature, whereas randomized sampling is a very practical way to proceed with characterizing solution spaces. And then there are a branch here of biased methods that bifurcates into many, many sub-branches, and this is where we will spend most of our time in part 3 of the class, is to study some of these branches. So that's a little bit of framing of this lecture and the transition we're about to go to from part 2 to part 3 of the class. Okay, so this is an illustration of these two unbiased ways of characterizing solution spaces. So there's an example, a solution space shown with a blue swath. And we talked about getting edge vectors, extreme vectors that turn out to be pathways last time. And we talked about how to get the minimum set of them and the issues associated with computing them and so forth. And we talked about the fact that every point in this space, such as the one shown, will be a non-negative linear combination of these edge vectors. So the weights on the edge vectors are either 0 or positive, but they're not negative. So that's one way to characterize the space and its contents, but we can also do something else, which is to sample randomly points throughout that space and get a sample, a statistically meaningful sample of all candidate solutions in that space. And then we can characterize these solutions to study the properties of the space. And that's what we're going to be talking about today. So the outline here is very simple. We're going to talk about 2 different approaches to doing sampling. One is how you sample low dimensional spaces, so maybe 10, 12, even 15 dimensional spaces. And then there are methods to sample high-dimensional spaces. The approach for the low-dimensional spaces is a little easier to understand conceptually, so I talk about that a little bit. But sampling in high dimensions is a very sophisticated procedure algorithmically and theoretically, so we'll just mention those characteristics. If you really want to learn that fully, that's an advanced topic that you could pursue through separate classes on that issue. Then the other half of the lecture is going to be on the uses of sampling. And I think you will see at the end of the lecture, when we've gone through these examples, that sampling is a very practical way to look at solution spaces, analyze data, and so forth. And I believe the sampling tools that are available in the COBRA Toolbox, I think even in the MASS Toolbox, certainly COBRA Pi, are relatively easy to implement once you have a model and a set of constraining data sets. And at the end, I want to talk a little bit about regulation, rotating basis vectors, and using samplings to determine the dimensionality of a regulatory problem. And it's a little bit of a different view of regulation. It's a network level view of regulation, namely how many things do you regulate to find a point in the solution space and drive the operation to that point. It's not the classical view of regulation when we're thinking about binding sites on an allosteric enzyme or a transcription factor binding, which are the individual molecular events. This is a network level view of regulation that helps you understand what the magnitude of a regulatory problem is for a cell to achieve a certain phenotype as represented by a point in a solution space. Okay, so let's first spend some time to talk about the thought process and the procedures that have been used to sample low-dimensional solution spaces. So this procedure basically has 3 steps to it. First, we define all of our linear constraints on a network, the equality constraints and the inequality constraints, and we've already spoken about these. Then, under these constraints, you sample the space uniformly. So the points have to be uniformly spaced throughout the domain of the solution space to give you a good, accurate view of what it looks like. And what you can then do is you can post-process that set of points by segmenting it into groups, by maybe imposing new constraints and throwing the points out that don't meet those new constraints. And that's kind of indicated in those panels to the left, where I have a red curve and looks like a blue curve, where the blue curve represents sampling of the entire solution space. Then I put in an additional constraint, and I look at only the sample points that satisfy that constraint, and I get these red curves. And you see in some cases there may be a big difference between these 2 sampled sets, suggesting that that particular flux will be quite different on average in the smaller space than the full space, whereas there could be other fluxes where it makes no difference whether you're adding that constraint or not. So this is the sort of thing we're going to be talking about. So let's first start by looking at this odd-shaped object here. What is the area of this object? How do you figure out the area of this object? You've probably all had your calculus classes where you go through the tables of solutions to integrals of equations, and there are whole books available of this. And I think it was after World War II rather than World War I, where there were a lot of unemployed mathematicians that actually worked day and night, week after week, month after month, year after year to generate these tables and solve these integrals. But of course, there are many integrals you can't solve analytically. So how would you figure out those integrals? And the approach developed to that was kind of interesting. It said, take that area you're interested in, engulf it with a space whose area you can easily calculate, like a rectangle, and then randomly and uniformly sample that rectangle and determine what fraction of those points are within the boundaries of your area and which are not. And then you take that fraction and say, okay, well, I know the whole area and I know what fraction of that area that object occupies, and that's how I'm going to calculate the area. It's a numerical procedure. You couldn't do it analytically. Okay, so basically, to get the size of this blue odd-shaped blue area is that you take the area of the box that you put it into, get the hit fraction of the points, multiply the 2 together, and you have your area. So you keep those points. So where does this leave us? This leaves us that we would like to generate a space that is easy to get randomized sample points in. And a square is very easy. You just randomize the weight on each one of the axes there and sum them up, and then you get all these red points. And also that space should be easy to compute in terms of its volume. You need to get the total size and generate these points. So let's talk about that a little bit. So the solution spaces we are looking at exist at high dimensions, and they can have very odd shapes, like the one shown over here. This is a very long and narrow space. So I would like to put a box around it that fits it pretty well. So here's a box that is probably not a good choice because only a small fraction of that box represents that blue area. And I might choose to redraw the box like this, and now I have a much bigger fraction of the box being represented by the blue area, and this would be easier to sample because the hit fraction would be higher, and you would get your statistics more quickly in terms of determining that area. Okay, so therefore, it is important to choose the smallest bounding area around these high-dimensional polytopes that allows you to sample and calculate the volume of the outer area, sample, get the fraction, and that'll get you the space and its characteristics that you like. And I will say right away that the shapes you're going to pick here allow you to compute the volume, which is an interesting property to be able to calculate. But it's more a question of being able to sample the space and figure out the points that are inside the space. When we get to the more sophisticated mechanisms, sampling methods, we're going to have to give in on the ability to compute the volume. But for this smaller dimensions, you can do that. Okay, so here is an outline of how this is processed. You start stating your constraints that we've been talking about quite a bit, the equality constraints and the inequality constraints. That may define that solution space shown here in this blue-greenish color. So we are interested in determining the hypervolume that satisfies all these constraints. So that's basically what we want to do. So the algorithm that is used to do this, or one algorithm that's used to do this, first you map all the inequality constraints into the null space, because the null space is defined by the equations, by the equalities that we have. So you map them all in there. And then you remove the redundant constraints. So we talked conceptually about dominant and redundant constraints, and there is a way to distinguish between them mathematically. And of course, the redundant ones are of no interest because they're not going to confine your space tightly in any way. So then you choose a subset of these constraints, and as we talked about, the maximum and minimum constraints on a flux form a parallel set of plates, and you have to be within them. Then it could be another set of max minimum constraints that intersects those two, and then so on and so on. And you end up with what's called a parallel pipette by just confining the space just by the max and the min constraints. They may not be independent, like we talked about, where the combination of the weights on some of the basis vectors are restricted. We talked about that last time. So with that, you can actually make many possible parallel pipettes, and you would like to calculate the one that is the most snug or the most tight around that space. And so here's an example of one parallel pipette you can draw based on set of max and min constraints, for this case that we're looking at here. And then here's another one that's more snug, and this would be a better one because it more tightly engulfs the space of interest to us. So yeah, there is an algorithm that can find the dominant constraints, look at all these parallel pipettes very quickly and figure out which one is the most snug. So that's done algorithmically, and we're not going to talk about that here. And then what you do, here is the more snug parallel pipette, and now we can sample that. And we get a bunch of points in there, some that are in the blue area and some that are not. So that's the sampling procedure. Then we just look at every point and decide whether it meets all the constraints or not, and that's not too hard either. And then we go ahead and we calculate the volume of the solution space and all the points that are in it, more importantly, that sample of points, by looking at the hit fraction. The hit fraction, of course, are the points we would like to look at, but we can also calculate the volume of the space by the equation shown at the bottom there. Okay, so let's talk a little bit more about this. So that's the outline, that's the conceptual background. So here is the case that we looked at in detail last time, the simple flux split with a flow into a node and two flows out, and each flux is non-negative and has a maximum bound as indicated. And we looked at this last time too, and we decided when we draw this out that there is that odd-shaped two-dimensional space there that we're interested in. All the points on that space satisfy all the constraints. And we also looked at this in 2 dimensions, like shown here. And I made a point of the upper right-hand corner of that space. If you extend the 2 basis vectors all the way, you end up in that upper vertex there, but that's outside the space because it violates the constraints on V3, which cuts through the space, and we talked a little bit about that last time. The way we randomize this space is that we take the max and the min value of each one of the alphas, and you can just normalize that to unity. Then you can get a random number generator on each one of those weights. And then you convert them into the absolute value as a fraction of that length, and then you add them up and you get a point in the space. And you do this over and over and over again for as many points as you'd like to get. And you get all the points that are shown in there and in that parallel pipette, and the black ones will be in the space, and the white ones that are in that little triangle to the top are outside the space. So we can calculate the area in this case and get the total size of the space, which is sometimes useful. The histograms there, blue ones to the far right, can be plotted for each one of the fluxes. So I just take all these points, and they're vectors with 3 values in it, and I can take all the values for V1 and I can compute a histogram. That's what's shown there. And the same thing for the second and the same thing for the third. So I can look at the distribution of these fluxes that are in there. And I can look at this a little bit more. So let's take V3. So here I'm showing with the green bar the minimum value, and is that the maroon color on the right, that is the maximum value. And all the samples have to be within there, and you can see that in the clearly when you just look in between these parallel planes. But the height of the histogram, and I show one example with the red bar, is really a measure of how wide the space is in between these parallel planes. So if you're moving between them, that width can get bigger or smaller. And you can just see that the histogram really represents the depth of the space in that direction. And you can see if I move all the way to the lowest value, the two basis vectors converge to 0. So having V max at 0 is the least likely value in that histogram, as an example. Okay, so that's the idea behind sampling spaces of reasonable sizes, or reasonable dimensions, I should say. Now, what are the limitations of this? The limitations of this is that it gets harder and harder to confine a space within a defined outer space as the dimensions go up. And the area that confined space occupies in the outer space gets smaller and smaller and smaller with every dimension. And that's kind of easiest to illustrate on how you fit a sphere, a hypersphere, into a hyperbox, or a circle into a rectangle, I should say a square, in 2 dimensions. And the formulas for the volume of a hypersquare and a hypersphere are easy to get, and there they are. And then you can ratio these numbers to figure out what fraction of the volume of that outer box, or cube, I guess it is, equal in all dimensions, what fraction of that hypercube is occupied by this sphere. And you can just calculate that. And as you might expect, as the dimension goes up, that drops very, very fast. And so fast that it's better to plot this on a logarithmic scale, which is shown down there. And once you start hitting 10, 12, 14 dimensions, this drops below 1 million. So 1/1,000,000th, I should say. So that means that on average, when you get to those dimensions, that you have to do 1 million samples before you get 1 point in the space. And when you start getting to those dimensions, it gets kind of impractical. But for volumes lower than that, if you have a big enough computer, you can sample spaces of that dimension reasonably efficiently, get the volume of the space, and get a good set of points that are in that hit fraction. But this becomes impractical when you go to 50 dimensions or 100 dimensions. Okay, so when you get to those higher dimensions, there is another way to sample spaces. And this is a pretty well-developed area of applied mathematics and numerics, is to be able to do this. And it's done by a Markov-Chain Monte Carlo type sampling methods. And I'm just going to show them to you conceptually, because if you really want to learn this, it'll take much more than 15 minutes in a lecture. This is a sophisticated subject, both numerically and algorithmically. But here's the idea. You have a space that is shown by this three-sided pyramid in 3 dimensions, and you have to find a point in the space. There are many ways of finding it. You can do linear programming, as we'll talk about, to find some of the vertices, and then you can average them and go into the middle of the space. And in fact, you can get many, many starting points in a space, and you can parallelize this algorithm actually as you search the space. And basically the way this works is that you randomize the direction in which you want to move away from that point, and then you randomize the distance, and you just keep walking around in that space. And there are certain criteria, numerical criteria, that will tell you how good your coverage is of the space and how uniform your sampling is and when you have to stop sampling. For a very oblong space, so this is almost like a diffusion process. So if you have a very long space and you see here in the end, it's going to be bouncing around there for a long time before it gets to the other end. So there are ways to deal with that. Like I said, you can do multiple seating points and sample in parallel. There are also ways to scale the space so it becomes more like a square than a long, rectangle. So that you can deal with that. So there is a long history of doing this. There are many variations of this algorithm that exist, and which one you use may be different depending on the characteristics of the space you have. So there are criteria for convergence to a uniformity, and there are better ways to get there quicker than others. So there's different ways of approaching this. And you have to set parameters when you do these things, like how do you choose the random direction. And it doesn't save every point because sometimes it's walking around for a while before you pick a sample point. So you can be generating many, many more points than the sample points you keep. So there are a few parameters you set, but you can sample high-dimensional spaces in this way. And like I said, this is an advanced topic that we will not talk about in detail. There's a whole book on this cited here at the bottom of the slide, and a lot of material available on how to do this. So what I would rather like to do is to spend our time to talk about what you use this for and how you gain biological insight, what kind of questions you can address, what kind of answers you get. So there are 6 examples I'd like to walk through, and I think most of these are in the text. And at the end I'll show you there is a review on the uses of these methods in this field. So let's start by talking about the human red blood cell. This has been a popular model in systems biology because it's the simplest human cell. And there have been models available of it for some time, and when you compute things for it, you get normally interpretable results. So here is an example of what you get. So in this case, you have the red cell, you have the model, the network, you have the reconstruction, and you know the uptake rates and the secretion rates typically for a cell like this, the glucose uptake rate and so forth. And you put those as constraints. You say, this is my input. You can give an error bar on it, say it's within this range, but the input has to be within this range, and then you sample the space. And then you can prepare histograms for every flux in the network, and this is what you get. So you see, for instance, a nice series of essentially normal distributions in lower glycolysis. You see in some cases the distributions are very flat, which means that every flux is equally likely. So you obviously don't know much about those. You have to get more constraints to get a better estimate of what the likeliest value of that flux is. Some are slanted to the upper end, so the high flux is the most likely. Some are slanted to the lower end, and so forth. So there are different distributions for each one of them. And when you show them this way, you're assuming them to be independent, but they are not independent. If you had sliders and you could cut out a certain value of one of the distributions, all of the other ones will change too. So if you restrict one flux range, say by a measurement, all the other ones change too. So these are not independent, and that's very important. So let me show an example of that. So when we talked about the basis vectors, I told you that you can use the Vmaxes as a slider almost to shrink and swell a solution space along the axis that corresponds to that enzyme. So if there's an inborn error in that enzyme, you can pull that slider down by whatever fraction the enzymes are reported to have lost in activity. So if they're at 10% of their, the nominal value in the human population, you move that down to 10% of that value, and now your space is much smaller, and now you can look at that reduced space and the statistical properties of the fluxes and see how they change relative to what you would call a normal situation. So here's an example of that. So glucose 6-phosphate, of course, represents an important branch point in the basic metabolic pathways. Either glucose 6-phosphate goes down glycolysis or it goes into the pentose pathway. So here's a set of the histograms from the nominal case that I just showed you, and I'm just showing a few fluxes in here. I'm showing pyruvate kinase at the bottom of glycolysis that has that nice normal distribution, and you have a most likely value there clearly. You see glucose 6-phosphate dehydrogenase that goes into the pentose pathway has a very different distribution. It's slanted to the lower value and goes down. So, and then you have hexokinase coming in. And often you put the zero flux as a red bar on these histograms because you're looking at so many of them, you want to know if it's positive or negative and how far away from zero the flux is predicted to be. So you see with the pentose pathway, it can hit zero. In fact, that's the most likely value in this case. Whereas the hexokinase and the pyruvate kinase flux are always of a finite value. Now, there are two very common defects in red cell enzymes in the human population. One is in glucose 6-phosphate dehydrogenase, and that's basically been selected for in the human population as a result of malaria and resistance to malaria. That shortens the lifetime of the red cell and therefore doesn't give the pathogen enough incubation time in the red cell. So it's very interesting how many variants there are there and how many people in the world actually have some issues with glucose 6-phosphate dehydrogenase as a result of surviving this nasty pathogen. The second one that is very common is in pyruvate kinase. And now I can restrict the flux through pyruvate kinase, as shown in that black box. I impose a maximum value that corresponds to that, and that cuts out a part of that distribution. So I form now a subspace where only a subset of my sample points will satisfy. And you see for now, both for the uptake rate and for the branch into the pentose pathway, the distribution changes markedly. The one through the pentose pathway becomes flat, okay? But these are not independent fluxes, like I said. And here I'm showing a two-dimensional histogram of the fluxes through hexokinase and through glucose 6-phosphate dehydrogenase. And in one case, if you look at the correlation through that two-dimensional histogram, there's almost no correlation between the two. And this is the normal case. So these things can be independently regulated just from the flexibility, stoichiometric flexibility is there in the space to move the point around. But look what happens to the distribution when you cut down the maximum pyruvate kinase flux. All of a sudden, you get this two-dimensional histogram where the hexokinase and whatever the flux is into the pentose pathway are not independent. So whatever the regulatory action the red cell is trying to take, it's hard to overcome because these fluxes become coupled just by the stoichiometric nature of the network and the prevailing inputs into it. And this is pretty important because just the structure of the system gives you that property. And there is some analysis of data possible here where you can look at how severe that restriction on the pyruvate kinase is. There are some data there from different patients groups. Group 1 is the one that has the most serious restriction where the size of the space is shrunk to below 2% of its initial size. and patients that fell into that category experienced anemia. So that's a prediction of how the physiological state of this cell is perturbed simply by mapping minimal information actually about inborn errors in metabolism. Okay, so let's continue to play with this a little bit, and let's look at the human mitochondria. As you probably hear over and over about in the popular press, this is the powerhouse of the cell, and any defects with it cause all sorts of problems. Experts in the field say that you're just as old as your mitochondria because the DNA in the mitochondria does get damaged with time, and it doesn't have as good repair mechanisms as the nucleus does. And this is mostly due to ROS species, so take your antioxidants and make sure that if you can minimize this process. Okay, so here is a reconstruction of mitochondrial metabolism in cardiac myocytes. This was generated based on some of the early proteomic profiles of the proteome in the of the mitochondria in cardiac myocytes. So you have that reconstruction, and there is data available on what typical uptake rates are into this organelle. This here is a sampling of the outer space, solution space associated with this organelle. And there are some physiological parameters put in here, but you can look at if these histograms are consistent with subsequent measurements that you get for activity of individual enzymes. And these red arrows show measurements of fluxes that were predicted to be in these ranges. And you see in most of these distributions, the measured flux is close to the peak, except the one in the bottom right, where the measurements is in that tail. It's an unlikely value for that flux. And remember that if you have measurements like this that are obtained after sampling, you can now use these measurements, just the nominal value plus minus the error, to shrink down these distributions and subset all the other, the samples for all the other fluxes, because this is information on how you constrain the space. So we can look at now variations in that space based on altered Vmaxes. And I may have shown this diagram before, but let me show it again. Here is a two-dimensional space, two fluxes that have been sampled, and we have these blue distributions. Now, if you have, for some reason, overactivity of V2, and its minimum value is elevated, that green swath disappears from that distribution, and that represents that green triangle in the solution space. And similarly, if you have the Vmax of the other enzyme go down, you also subset that solution space. So this is what you can do. So you can put in to this outer sample set, you can put in conditions for a certain pathological state, like diabetes. You can put in different inputs into the mitochondria based on nutrition. You can put in inborn errors in there. You can cut off oxygen, like if you have an ischemic event in the heart. So you can study all these perturbations to the flux state of the mitochondria in cardiac myocytes. And this one diagram here shows the subsetting of the outer solution space based on numbers obtained from diabetics. And there are a few noteworthy things in here, including this diagram that I'm going to talk about in a moment. You see that many of the red histograms become very narrow. So the operation of metabolism in the mitochondria in the heart of diabetics is expected to be much less flexible than in somebody that's considered normal. It kind of gets more restricted once you put those constraints on the network. Oxygen uptake rate is restricted in particular to gets closer to the maximum. Interesting thing here is that pyruvate dehydrogenase in the mitochondria is shown or highlighted in that box. Is that a maroon color? A maroon colored box. And you see that the red histogram gets squished up close to zero. And this is a known phenomenon apparently in diabetics, that the flux through this enzyme is very small. And people have thought that that was a consequence of altered regulation. But this analysis would say this is a network property. If you change the inputs and so forth, the only way to balance the map is to drive this flux to a very low value. So not a druggable target, in other words. It predicts increased lipid synthesis, which of course you see in diabetics. And in this paper that is cited there, other perturbations were studied, like cutting off the oxygen. What happens when you cut off the oxygen? And if you provide different inputs, high lipid diet, low lipid diet, and so forth. So this is what you can do, very practically. Just reconstruct, in this case based on proteomic profiling, you build your map, you sample all these candidate states, and then you start looking at the inputs and the constraints that are placed on this network under different conditions. All right, so let's talk a little bit about the flux map of E. coli. One of the very early papers in this, with this approach was published by Laszlo Barabasi's group when he was at Notre Dame, and this paper was published in Nature a little over 10 years ago. And this was really a shocking revelation to me and others in the field that you could do this. But this is apparently a common procedure in physics, which is the background of that lab. But they just started sampling solution spaces, and this opened up a whole new dimension in COBRA tool development. And basically what these diagrams show, and the one in the top left in particular, is that if you just sample the solution space and you take all the fluxes in every vector and you bin them and you look at the distribution, that there's a power law distribution of flux loads through metabolic reactions. So this was the first indication that the power law properties of the topological features of a network that we spoke about a few lectures ago seems to be preserved in the activity state of the network. And the panel to the lower right is actually experimental data from Uwe Sauer's lab at the ETH that had been published at that time, and it also shows this kind of power law distribution. So this is, a way to characterize the solution space. And it suggests that the enzymes, the way they're used, the way they're loaded with flux, follows a power law distribution. If you just sample all the candidate flux states, which is done in here, or even the optimal flux states. Okay, so here's another example, and we're going to talk about much more about this later in the class. I think as some of you know, we can now do evolutions in the laboratory and watch how the physiological state of a cell migrates from a suboptimal state to an optimal state that corresponds to the selection pressure. So the whole state of the metabolic network migrates, it shifts. So you can now study, if you know the inputs and outputs, during the adaptation or during the evolution, you can make predictions about which fluxes are going up and down in their value. So the top panel there to the right shows growth rate improvement over time, and that dashed vertical bar is 40 days, and it seems like after 40 days in this experiment, the growth rate stabilizes and is not improved after that time. The red curve on top is done at a higher temperature. And what's shown at the bottom is something that we're going to get to later. These are called phase planes, and they show the substrate uptake rate on the x-axis and the oxygen uptake rate on the y-axis, so the two primary inputs. And the first panel of those, in the top left corner of these box of 4 panels, shows a bunch of points that are obtained for the wild type under different conditions. And the panel to the right of that has that trajectory in it, shows the measurements of these uptake rates during the evolution. And you see them migrate to the red diagonal line, which is the predicted endpoint of that experiment. And we'll get to it later. And the 2 panels at the bottom shows various measurements of the strain after it reaches that optimum, and it just seems to stay on that line. So we'll talk about this later. This is an example of how you can predict an optimal growth state and produce it by laboratory adaptive evolution. Okay, so this is done on glycerol. So the wild type strain grows poorly on glycerol. You can evolve it. It takes about 700 generations in this case to double the growth rate and reach that line. And the adaptation is reproducible with different experimentation. Now, what we can do is we can take these measurements of the uptake rates and secretion that are put on the network, and we know the growth rate. And we can, with those constraints, you can sample all the key fluxes in metabolism. And you see these multicolored histograms that show the predicted flux value that is consistent with those physiological measurements at different points during the adaptation. So some fluxes are predicted really to change significantly. Others you can't tell anything because the distributions look essentially the same. So this might be a guide to knock out those genes in the evolved strain, for instance, to see if those that really go to high value are very consequential and so forth. So you can do experiments with this now to interrogate this predicted most likely flux map as a result of the adaptation. So this is moving a little bit more towards prospective experimentation as a result of a sampling process. So this success with using just the uptake rates led to a much more detailed examination of what you could do with the exometabolome. And why is that important? Now it turns out that it is very hard to measure intracellular metabolites and intracellular fluxes because you have to rip the cell apart, and as you rip it apart, the metabolites are labeled, they are labile, and they may change concentrations very fast during your separation process. They have to come off the enzymes where they are maybe naturally kept in an intact cell and so forth. So this is hard. However, the media outside the cell is much easier to access and monitor. And normally the metabolites are outside, are stable. You can harvest the media, and you can measure quite precisely the concentration levels outside the cell as a function of time, and you just take the slope of that curve, and that tells you what the uptake or the secretion rates are. So those are things you can get pretty easily experimentally. How informative are those measurements if you can map them into such a sampling procedure? Okay, so measuring is easy. And the changes in the uptake rates can be used to constrain the solution space, and you can put them into a workflow. And this example, I chose this example because it shows you that this sampling procedure becomes a part of a workflow. I think I mentioned this before, that in this field, one measurement, measurement method or approach will answer everything. No computational approach will answer everything. You have to string them together into a workflow to get to the answer that you want. So here's an example of such a workflow. This is the work of Monica Moe. So in panel A, it shows the secretion rates. You run your mass spec in that case to get the concentrations. Then you take the slopes and you use them as constraints on the inputs and outputs. And that forms a solution space for you that's consistent with those inputs and outputs. And then you can sample that. And you can sample that under one or two conditions. And what's shown in panel E, in the middle of the bottom row, are then all these histograms that you may get for the same network under two conditions. So she studied in particular a yeast cell that had where a gene had been removed, and she knew the answer because the gene had been removed and that flux went to zero. But that was a test case for her, and she was able to show by this analysis that that was one of the most likely perturbations intracellularly. Now, just a little bit on how this is approached. So here we show two sampled spaces, blue and red, and you measure maybe the distances between points, all pairwise combinations in these two spaces, and you generate a histogram. Then you generate a z-score that you use for statistical purposes that you can use to define what's called a reporter metabolite, maybe the nodes in the network that had the most varied fluxes around it, and that if you measure that will be the most informative measurement. This is something developed by Jens Nielsen's lab Chalmers, and so forth. So this is a very practical use of sampling to do informative data analysis. So can you use this can you use metabolomic measurements to constrain the solution space? The answer to that is yes. You can use that to compare multiple flux distributions. And there are slightly different ways of doing that statistically. In Monica's study, she had knockout strains that she could analyze. But as you might imagine, this has been used for many other applications, important errors in metabolism in particular. Tomer Shlomi in Israel has used this to predict biomarkers. You can impose the inborn error like we did in the red cell case before. That would predict for you which fluxes on the outside are likeliest to change. And they might represent biomarkers for you. So that's another example. And there are a few variations of this kind of workflow that have been developed, and their uses are many. Okay. So that's an example of a workflow and how statistical sampling and data mapping can be useful to get at an answer. So let me now talk about an analysis of brain metabolism. And this was done by Nathan Lewis, who is here in the pediatrics department now. So Alzheimer's, as I think you all know, is a pretty nasty disease. I hope nobody in the room will ever experience this, or their relatives, because this is a very taxing disease. And a lot of money is being spent on studying it, and as with many malfunctions of the brain, there is heavy metabolic involvement in these malfunctions. Whether they're causative or whether they are a consequence of something else is a different matter. Although people are beginning to hypothesize more and more that at least with neurodegenerative diseases, that metabolism may be causative and related to the loss of redox potential in the ER, which leads to malfolding of secreted protein. But anyway, in Alzheimer's, apparently one of the hallmarks of the disease are changes in the activity level of some enzymes, including alpha-ketoglutarate dehydrogenase, pyruvate dehydrogenase we talked about before, and a certain cytochrome. So the activity levels of those changes apparently markedly in this disease. And this happens in all the neuron types. There are three principal types of neurons, as we get to in a moment. Well, right here. So the cell death in Alzheimer's is confined apparently to two types of neurons. These glutamergic and cholinergic neurons that apparently deplete first, but the GABAergic neurons apparently persist. And one of the questions in the field is, why does that happen? Why are they somehow neuroprotective, for lack of a different word? So Nathan collected data sets on these neurons and on this disease and came up with this paper here that was published in Nature Biotech some years ago. So in these first few panels, he has sampled the solution spaces for these different types of neurons. And on top, he has the CO2 release from metabolism that apparently can be measured. In the following three panels is the predicted level of activity of the cytochrome. And the neurons are color-coded. So the top ones you see in Alzheimer's, the histogram, the dashed lines, which is the disease states, and the solid line is the normal state. So there's a big difference in that prediction for the two types of neurons that are depleted. Whereas the GABAergic, you can see that the histograms are essentially the same. And down below, we have predictions of flux through the GABA shunt. And you can see that in the GABAergic neurons, that seems to stay in the normal range, whereas in the other ones it seems to go down. So this pathway map shows the GABA shunt in the middle of that cycle, and to the top in the light blue is the alpha-ketoglutarate dehydrogenase that seems to be reduced in activity. In the panels below here are the expressions of alpha-ketoglutarate dehydrogenase in the different regions of the brain. And I'm going to box in here two of the regions that are mostly affected by Alzheimer's. And this analysis points to two genes, GAD genes as I recall, that are selectively upregulated in the GABAergic neurons and affect that flux map. So there is a hypothesis that comes out of this, which is that those genes and their involvements in metabolism are important in conferring protection on GABAergic neurons. So that's a very complicated situation. A lot of data goes into this analysis, but in the end, the sampling is the key to analyzing the data types and reaching the conclusions that I just stated. Okay. Yeah. So these are basically the conclusions, that the neuroprotective effect of GABA is specific to GABAergic neurons and is presumably related to a couple of genes, actually 3 genes that were fingered as a result of this analysis. So the pathology of Alzheimer's is of course more complicated, but at least there is some consistency there with the metabolic property. And I would say that this study here is important from the standpoint that this is the first one that really analyzed subtasking of metabolism in the tissue environment in humans. I didn't mention that there are astrocytes involved in this, and there are cells around the neurons that support the neurons metabolically, that play a support role. And this all comes down to exchanging metabolic tasks and loads between these cells. And this is true in most microenvironments in the body, like in the tubule in the nephron and so forth. So there are different cell types in there, and they have different metabolic roles, and they need to be perfectly synchronized to get the function of that, what is called a functional subunit within a tissue. Okay, so the last example I want to talk about relates to analyzing the interaction between hosts and a pathogen. So what is shown here is a lung macrophage to the top left, and a pathogen, an intracellular pathogen, TB, that infects lung macrophages. And a model for TB has been built, it's been reconstructed and so forth and studied by Nima Jamshidi, who is a radiologist now at UCLA. And you can take Recon-1 and customize it to the lung macrophage based on expression profiling in it. And interestingly enough, you can also have an infection model in vitro, from what I understand, of the pathogen going into the macrophage, and you can expression profile them separated and together, because you can pick out the bacterial transcripts and the human transcripts separately and map them separately onto the models. So you can start to look at the metabolic interactions between them. And the question is, does the infection change the metabolism of the host? Does it change the metabolism of the pathogen? And can we use anything based on this? Can we sample the potential states and learn anything from them? So the answer to that is yes, otherwise I wouldn't be talking about this. So this here diagram shows the core metabolic pathways in the pathogen when it is outside the host and inside the host. And the blue histograms in these callouts here are the host, if the pathogen without the host, and the red ones are within the host. And you can see there are notable predictions of shifts in metabolic pathway. So the glyoxylate shunt here is to the bottom left. In the middle there is enolase and glycolysis, where the flux is essentially predicted to go to 0 in the affected state, so no glycolysis. And FBP is a gluconeogenic enzyme that goes opposite to phosphofructokinase, and that is predicted to be very active in the pathogen when it comes into the host. And in fact, the pathogen is predicted to consume fats and glycerol once it's inside the host, and have these shifts in its metabolic state based on the expression profiling data that has been obtained and mapped onto the reconstruction. So that's very interesting. There's a dramatic shift predicted post-infection in the metabolic state of the pathogen. So on the host side, there is a study that's been done that is too complicated to detail here, but macrophage metabolism has characteristics relative to it being activated or suppressed. And in fact, when the immune system mounts a response, the macrophages actually communicate quite closely with the adipocytes to basically see if there's enough energy to mount a response of what magnitude. And in the box there, A, with the reddish background, are pathways that are known to be associated with activation, and the blue boxes are metabolic pathways associated with the suppressed state. And what's shown in this diagram are some highlighted red circles, and these are the reporter metabolites that I mentioned earlier that are likely to be very informative in the system. And if they are engulfed by a circle in this diagram, it means that the measurements that were done in the resting macrophage versus the activated macrophage were significant when the measurement was done. And these, the multicolored bar there to the right shows the model prediction labeled MP versus the data, and the red ones are the predicted to be activated fluxes, and the blue ones are the ones predicted to be suppressed. And this is from an experiment where a macrophage is exposed to lipopolysaccharide, which really inflames macrophages. So this model here, this analysis of the host, distinguishes between the activated and the suppressed state, and is validated by these measurements. Now, what I'm going to tell you, and not going to show you anything about, is the fact that when the pathogen, TB, goes into a macrophage, it actually promotes the suppressed metabolic state. That's how it affects the macrophage. It keeps it quiet. Which is an interesting result here. All right. So some points here. We can change the shape of a solution space based on data, and if you sample the solution space, we can re-estimate the most likely state of the fluxes. In this case, this last case, we look at the fact that the separate cells, the host and the pathogen, operate quite differently than them together. And we see that the pathogen seems to promote a suppressed state in the host. And the pathogen itself undergoes a massive shift in its metabolic state after infection. At least that's the prediction. All right, so this is a series of examples of increasing complexity and biological depth that I've run through just to show you how useful the sampling procedure is. It's very practical, it's easy to implement, you can get the answers pretty easily, and you can use it for data analysis. In contrast, the extreme pathways are tougher to use in this regard, but there you can maybe view them as being more fundamental in terms of the way they characterize a solution space. Now let me end here by talking a few minutes about how to address a conceptual or a theoretical issue using sampling. And we can use the study of dimensionality of solution spaces to interpret regulation and the network level point of view of regulatory needs. And this comes down to directions in a solution space and its dimensionality. Because if you want to locate yourself in a space, you have to be able to control its dimensions. And each dimension in the space we are talking about here is a set of fluxes. So if we can regulate those fluxes, we can locate ourselves in that solution space. Okay, so we can map onto a network known regulatory rules. So if you have a certain condition, the cell will sense that, implement the transcription factor, up and down regulate genes. And we can use that to subset an outer solution space based on observed regulation. That'll give us a smaller solution space, but not the point. And then we can sample that smaller solution space and look at its dimensions and try to infer what the needs are to regulate the gene product activity of upregulated enzymes to get to the state that, the eventual state that the cell takes on. So this is then a two-level problem. One is to look at the transcriptional regulatory network, and it is often described with a hierarchical image like shown to the top left, where at the top of that hierarchy are the global transcription factors, then the more local transcription factors, and then eventually the genes that are regulated by them. So global transcription factor may regulate another transcription factor that regulates a third one, and that third one has some target genes below it. That's kind of the way this hierarchy is. And as this network was known in 2004, you could take the E. coli network and put it under certain growth conditions and calculate the solution after, well, let me back up. You have a condition that you put the cell into, and there are 15,500 conditions of this sort that the E. coli network could respond to and could be represented by known transcriptional regulatory activity. So under each one of those conditions, you can customize the network by shutting genes off and on, and then you can look at, what the state of that system is. And that's what's shown with this little green, red image here in the bottom left, where the conditions is 15,000 of the columns, and the rows in here is the expression state of a gene consistent with that. And you can just look, so this is a huge matrix. It's like 15,000 by, I don't know, at that time, maybe 1,000 genes or something like that. But you look at that matrix and you can see that it's low dimensional. It has a very strong band structure. These columns are very similar. So you can do a dynamic, you can do basically a PCI type analysis of this and look at the dimensionality of this matrix and look at its main dimensions. And not surprisingly, it turns out that the biggest variation in that state is determined by what electron acceptor is available. The second biggest variation in the state is the carbon source, mostly glucose and non-glucose sources. And the third most prominent dimension was the nitrogen source, which is interesting because those three, availability of three nutrients has a dominant effect on what the metabolic state is. So this analysis says that with respect to the macronutrients and the major fluxes in a cell, it's really a fourth-dimensional regulatory problem. You can have redundancies and have more regulation, of course, but if you have less than 4, you're in trouble because then you have unregulated dimensions in your space. The space that is then closed off turns out to be effectively 7 to 8 dimensional. And this is the way you deal with studying that reduced space. What is shown in this series of events here, this is a workflow, is that you have a network, metabolic network, you have transcription factor activity, you get a space, you sample that space. You take that set of sample points and you do an SVD on it to look at the dimensionality of that space. And since SVD is dissatisfactory, like we talked about with respecting the stoichiometric structure of the network, you rotate that base minimally onto the pathways. And by doing that, you can align the dimensions, the dominant dimensions in the space onto the pathways. And in this case, it shows the pathway stubs that corresponds to these 7 or 8 dimensions, and these have to be regulated tightly. And these turn out to be major segments of the major metabolic pathways. So if you can have 8 regulatory events in here to specify the fluxes through these pathways, you can define where you are in that reduced space. So this is an interesting point of view of regulation. Once the space is clamped down by transcriptional regulation, just what is being expressed, it's a 7-dimensional space, and these 7 pathways shown in here account for almost all the variation in that space. unbiased method, and it gives the overall dimensionality of the regulatory problem. So I think it is important to know that this is coarse graining, gives you the overall regulatory challenge that an organism has. But for an organism to be perfect in its operations and perfectly fit, it has to also make sure it buttons up the rest of the 4% of the space to get, perfectly the right space. So it's not saying that the other regulations are unimportant. It needs to be there, but these are the dominant features of the regulatory problem that E. coli is faced with under the growth conditions studied here. All right, so just here at the end, what else can be studied by sampling? There's a lot of other things that can be studied by sampling. You can look at correlation between fluxes like we showed in that red cell example at the beginning. You can get quantitative predictions about disease states as we saw in the mitochondrial case. We've not talked about the prediction of therapeutic targets here, but people have done that. Similar sort of work has been done with dynamic states. We've only talked about steady states in here. And there are several studies out that show how this is used. And the reference there at the bottom shows a citation to a review that looked at, I forget now, 40 or 50 of the early studies in using this method. I thought I should add this also. This has come out since then. This is from Jens Nielsen's lab at Chalmers. Shows a workflow that includes sampling two spaces from two different conditions. And based on the properties of that space, there is a prediction as to whether a gene product is transcriptionally regulated, post-translationally regulated, or allosterically regulated. So you can classify the likely regulatory modes based on the regulatory needs of the network of the sort that I was talking about. So sampling can be used for a lot of purposes. And here are some summary bullet points. We have methods to randomly and uniformly sample solution spaces, the ones that we have defined by the constraints we impose. They can be fundamental, like the flux balances and absolute values on Vmax and Vmin, or they could be defined by data-driven constraints, like the optic rates that I talked so much about, or expression profiling. So there are two methods used for sampling. The one for low-dimensional spaces that was used quite a bit early on, and there's another one for high-dimensional spaces. And not surprisingly, that's the one that's predominantly used. So we can get a global view of the context, contents, and the features or characteristics of solution spaces in this way. We can look at these correlations between fluxes. We can segment the space based on additional constraints. We can subtract two spaces, like under two different conditions, and so forth. And so this is a versatile, easy-to-use Cobra tool that has numerous productive and practical applications. So that is the end of part 2 of this class, where we talk about the stoichiometric matrix and its properties. And we had these 2 lectures at the end that talk about the unbiased way of characterizing solution spaces. One is these extreme vectors or these extreme pathways that help us gain a fundamental understanding of a solution space. And the other is this uniform randomized sampling that really helps us quickly get an understanding of the solution space and its properties as a whole.